In parallel with our therapeutic development programs, InMed has been developing an integrated cannabinoid synthesis system to manufacture pharmaceutical-grade cannabinoids. Previously, we focused exclusively on biosynthesis, but our R&D efforts had led to IntegraSyn, which we think is a way to optimally manufacture cannabinoids at a very low cost. Today, I'll explain how IntegraSyn works and show you how the IntegraSyn manufacturing system is flexible and able to produce a wide range of cannabinoids. IntegraSyn is designed to be an efficient, scalable, flexible, and economical solution to produce cannabinoids with bioidentical structures to those found in nature. We believe IntegraSyn may be able to address the current limitations of traditional cannabinoid production processes such as extraction, chemical synthesis, and biosynthesis alone. So let's take a look at IntegraSyn and why we think this is the most cost-efficient way for cannabinoid production. IntegraSyn uses common, well-established pharmaceutical manufacturing processes such as biosynthesis, biotransformation, and purification. As with InMed's previous approach, it all starts with E. coli biosynthesis. In this case, we're not making a final cannabinoid through biosynthesis, but we are producing a high-efficiency enzyme. We can do this at a very large scale and at exceptionally low cost. This high-efficiency enzyme is then combined with other substrates or starting materials in what's referred to as an enzymatic biotransformation. The enzyme, combined with these other starting materials, actually manufactures the cannabinoid. This is called the upstream process, where the cannabinoid is actually made. This crude cannabinoid mixture is then further processed in what we call the downstream purification processes. This involves a number of steps of purification, separation, and drying in order to separate the cannabinoid from the reaction mixture. This leads to an API-grade cannabinoid, which stands for active pharmaceutical ingredient. This is something that could be used as a pharmaceutical product. This is one of the key advantages of IntegraSyn, is the flexibility this process affords. So you can stop with this API-grade cannabinoid, or you can use it for further processing into another cannabinoid. This can be done one of two ways. You can go back into the enzymatic biotransformation process using a different enzyme, or you can actually use chemical synthesis to further process this cannabinoid into a differentiated rare cannabinoid. Regardless of which path is chosen, the cannabinoid will then be further processed using the downstream purification processes, resulting again in a different API-grade cannabinoid. This process is flexible. We're able to differentiate a number of different cannabinoids using the same process. It's modular in that we can stop at several places along this process. We can inventory the product that's made and use it later on. It's very cost efficient. We use very cost efficient substrates in order to manufacture these cannabinoids, and it's scalable, anywhere from small scale to very large pharmaceutical scale. We believe this process is optimized to provide accessibility to rare cannabinoids at pharmaceutical quality.